1, 2, 3 o'clock, 4 o'clock, rock. 5, 6, 7 o'clock, 8 o'clock, rock.
It all starts with fertilization. Vertebrate embryonic development forms a blastula that continuously divides to eventually form a morula. The morula then secretes a fluid within it that forms the blastocyst cavity, also known as the blastocell. This development continues until the epiblast cells become distinct. These cells form the embryo. At the tail end of the epiblast cells, a primitive streak forms along the midline, with a primitive node just ahead. The node signals the cells at the primitive streak to start to divide and fold underneath the epiblast layers, forming the two new layers called the endoderm and the mesoderm. The epiblast layer is now referred to as the ectoderm. The node then signals a tube of cells to form under the ectoderm along the midline, called the notochord, which signals the formation of the neural tube by the ectoderm. As the neural tube forms, the mesoderm migrates into three different areas: the lateral plate mesoderm, the intermediate mesoderm, and the paraxial mesoderm, all of which run down the midline, mirrored at either side of the neural tube. The paraxial mesoderm is what segments to form somites. Somites drive the differentiation into the cartilage of vertebrae and ribs, muscles, tendons, and the cellular sheath of the spinal cord. The differentiation depends on the cells placed in the somite. This process is very tightly regulated by the increase and decrease of expression of certain genes called the wave and clock model. There are several transcription factors that regulate the segmentation clock, one of which includes the transcriptional repressors, SNAILs. SNAILs have been of particular interest to the Cell and Developmental Biology Research Unit of the University of Dundee.
Okay, so I'm sitting here with Dr. Kim Dale from Cell and Developmental Biology Department of University of Dundee, College of Life Sciences. Dr. Dale is the leading author of a paper in oscillation patterns of snail gene expression and how it affects embryonic development. So, Dr. Dale, would you first tell us a little bit about your lab and your research?
I'm a developmental biologist, which means that the focus of my lab is to understand how one goes from a single fertilized egg to a fully formed embryo where everything's developed at the right time and at the right place.
So, focusing on the paper itself now, what were your original hypotheses in the investigation?
We wanted to find whether or not we could find candidates that could potentially be linking the different pathways that we know are involved and crucial for somite formation and somatic. So during somite formation, it's a periodic process where somites are formed in a progressive manner from the head to the tail, and that periodicity is thought to be regulated by a molecular oscillator that we call the segmentation clock. Segmentation clock can be visualized through the dynamic gene expression both at the RNA and the protein level of genes that we call clock genes. And these clock genes are components of three specific pathways in the mouse, which are the Notch pathway, the Wnt pathway, and the FGF pathway. Those clock genes, the dynamic expression is thought to be regulated through feedback mechanisms, since the large majority of clock genes are negative regulators of the pathway that activate them. And as well as being negative regulators, those that have been looked at the protein level are also very unstable. So you have negative regulation by unstable inhibitors, so when the inhibitors are degraded, a new wave of transcription begins. But what had been done up to date was to basically look at those pathways in isolation, and one thing we wanted to know was whether we could see any forms of crosstalk at different levels of the transduction pathways. And so we started to look at these snail genes because in different developmental contexts they've been described to be downstream of the Wnt pathway or of the Notch pathway, and since those are two of the key pathways involved in somatogenesis, we thought it'd be an interesting point of investigation. So the other thing that happens during somatogenesis is that the pre-somatic mesoderm tissue that generates these segments or somites is initially a mesoderm tissue, and it becomes an epithelial sphere, which is the somite, and the snail genes have been shown to be critical in driving epithelial to mesenchymal transitions, and since the opposite is happening here, we thought it would be interesting to look at what the function of snail was.
Having retrieved your results and analyzing your findings, what can be taken from your investigation, and what implications do they have on developmental biology?
So firstly, we achieved the goal, which was to ask whether or not a snail acts as a point of crosstalk between these two pathways, and we find that it does, in that we see in the mouse mutants that lack components of the notch pathway, expression per se of snail is not affected, whereas in the transgenic lines lacking components of the Wnt pathway, we do see a complete abolition of the expression. So that tells us that regulation of the expression of snail is Wnt dependent. But nevertheless, when we misexpress snail and cause an overexpression, we do impact on the oscillations of the notch target genes, showing that these two pathways are talking to one another, and that snail forms an important hub, of that crosstalk. We also found, as we had expected, that there would be an impact on this epithelial mesenchymal transition. So snail maintains things in a mesenchymal state. So maturation and epithelial somite formation was disturbed through misexpression of snail. And that has sort of tied in and corroborated studies, and has probably led on to, a lot more investigation into the role of snail, not only in the presemitic mesoderm, but also in neural crest development, where there's epithelial mesenchymal transitions. Not only that, but also in disease states. So cancer obviously involves EMT and the opposite transition, MET, when cells are metastasizing and moving from the initial tumor site, metastasizing to a secondary tumor site, and snail has been implicated in those transition states. Is absolutely crucial for regulating EMT and MET. Findings and also the interactions between snail and other pathways that are found within this context can be picked up and then looked at and investigated in all of these other very, very important biological situations.
What steps has your department taken since the investigation? For example, is there any research currently that is in heavy ties with this investigation?
This paper was actually generated while I was a postdoc in my former lab, and that lab has gone on to generate mice lines that carry mutations in different snail genes to look and see whether, in the absence of snail, what the effect of that is, because in this paper we were only looking at misexpression. Those studies are ongoing at this time, at that point in time. But what we've done in my lab is to go on and ask, what the molecular regulation is directing the periodicity of somite formation. So can we get a molecular handle on what drives and regulates the pace and the periodicity of oscillations? And we've been focusing heavily on the Wnt pathway in that context, and we've generated data to show that Wnt is important in regulating the pace, and that's the direction that my lab's taken.
1, 2, 3 o'clock, 4 o'clock, rock. 5, 6, 7 o'clock, 8 o'clock, rock. 9, 10, 11